students. This is our first lecture in which we are going to discuss some of the basic principles of journal pharmacology. So before studying pharmacology, we should know what the term pharmacology means. So we can divide this term into two important parts, that is Pharmacon and logy. And logos. Logos, as usual, it means study of or science related to something, and pharmacon has come from a Greek word which means drug, and further drug has been derived from a French word which is drogue, which means dried herb. Now whenever we hear the term drug, one thing comes to our mind is that any substance of abuse or any substance of misuse. But in medical terms, drug has been defined as any substance or any product which is used in the diagnosis, treatment, or prevention of a disease. It can be any component which is used medically for diagnosis, treatment, or prevention. And how this drug or this chemical substance, when it is given from outside the body, that is any exogenous substance. Any exogenous substance, when is given to a living organism, how it is going to affect the processes of the body. How? What it is going to do, whether it is going to increase or decrease the hormone level, or it is going to affect your growth hormones, growth factors or growth hormones, or it is going to affect your neurotransmitters, etc. What effects are going to be produced in a living organism? That is pharmacology. So the interaction of the exogenous compound, exogenous chemical molecule with the living system to give a biological response which can be increase or decrease in the activity of the living system, that is a study or which is known as pharmacology. Now something regarding the history of pharmacology. There was one person from Germany which is Oswald Schmidberg, who is known as father of pharmacology. Why he is known as father of pharmacology? Because he was the one, obviously along with his disciples, he was the one who proposed the fundamental concepts of pharmacology. And there was his one student, that was John Jacobs Abel. He is known as father of modern American pharmacology. And he was the one who first isolated hormone. And that hormone was adrenaline from adrenal medulla. And this hormone was discovered in 1900 by John Jacob Abel. Now, being Indians, we should know the father of modern Indian pharmacology, that is Ram Nath Chopra. He was the one who first established the Center of Study and Research in Pharmacology in India, that was in Calcutta. This was the brief history of pharmacology. Now we should know why do we need to study pharmacology and what is the scope of pharmacology. So basically there is an undying need to discover medicines or to find out medicines, how to treat different diseases. So pharmacology has an undying scope. But to discover the medicines or to know how the medicines are going to react inside a body, first of all we should know what is the disease or how is the disease being caused. Disease is what? Disease is a some kind of dysfunctioning in the body. It is a dysfunctioning of some of the functions of the body, and how that dysfunctioning is being caused, that we should know so as to correct this dysfunctioning. And when we know what is the dysfunctioning, we will find a substance which is going to correct this dysfunctioning, and that will be our drug or our medication. For example, if we take a disease to be peptic ulcers, we know that in peptic ulcers what is happening, acid, the HCl in the stomach is being produced in higher quantities, and what we need to do is either we can have a symptomatic effect with the help of antacids, but that is just a symptomatic effect. It is just going to form a lining around the inner lining inside the stomach and prevent that acid to cause further damage. Or there are certain agents such as cimetidine or ranitidine, which are histamine blocker agents, and they are going to basically lower the HCl secretion. Other agents which are again going to lower the HCl secretions are your proton pump inhibitors. And for example, omeprazole is one example, pantoprazole is second example. So these are going to inhibit the HCl. So when we know that HCl is going to be increased in case of peptic ulcers, therefore we need to find agents which are going to reduce the HCl. So we know what is the dysfunctioning and we are finding ways to correct it. And this is the scope of pharmacology. This is the need of pharmacology, that in order to correct these diseases we need to know what is the basic problem in that disease, and we are finding ways to correct it. Okay, now from where these drugs are coming, what are the sources of these drugs? So these drugs can be obtained from different sources. There are certain animal sources. For example, we are obtaining insulin nowadays from animals. There are plant sources. We are obtaining digitalis. Opioids we are obtaining from plants. There are inorganic or mineral substances can be formed, and there are synthetic substances which can be produced in the laboratory. So once we have identified the source, we know that this plant is going to give us a certain drug, then what we are going to do next is we are going to identify the active ingredient inside that source. We are going to identify which ingredient among that source is acting as the active ingredient. After that, we need to check its pharmacodynamic and pharmacokinetic properties. Now, pharmacodynamic and pharmacokinetic, what are these? These are the two main branches of pharmacology which we'll be discussing in more detail. For time being, we will say pharmacodynamics is the biological effect which is to be produced by that drug, what effect that drug is going to produce on the body, and pharmacokinetics is what body will do to the drug. That is kinetics. The drug will be broken down, absorbed, distributed, metabolized, eliminated from the body. That is kinetics. What effect is it is going to produce in the living organism? That is dynamics. So after we have identified the active ingredient, we are going to check its dynamic and kinetic properties. Then we are going to ensure a precise constant dosage. At what dose it is giving a particular effect. Then we are going to check the structural relationship properties. For example, we have identified the active ingredient and we know its chemical structure. We modify that chemical structure in order to enhance the properties of the drug. That is your chemical alterations. Okay. After this, if the scientist is satisfied that this drug which you have obtained a drug, this is going to give a desired effect in the organism, he files this drug as a potent drug, or it can be used in the future. So he will do the necessary applications and everything, and then this drug goes into the drug developmental phase. So what is drug development? In drug development, there are two phases. There are preclinical testings and there are clinical testings or clinical trials. So in preclinical testing, what are we going to do is we check the drug for its biological effects. Initially, its biochemical and physiological investigations are done, the receptor binding assays are done, and then experiments on the cell cultures are done, or isolated organs are done. You isolate an organ, for example the heart or the intestine of the animal, and then you test your drugs on that isolated part, and if you are satisfied, then these tests are done on the living organisms, such as the in mice or in rats or in rabbits, in order to check what this drug will give that effect in the living organism. Because when you are checking the drug in an isolated organ, you are just checking in that particular organ, but you are not sure what that drug will cause if you are giving it in a living organism. If it is having a positive effect in a certain organ, it may have a negative effect on the other body parts. so you are not sure. You need to check it in the preclinical trials on the animals first. If your drug passes the preclinical trials, it has crossed all the toxicity testings, the genotoxicity testings, the carcinogenicity testing, the teratogenicity testing, then your drug passes this preclinical trials and they move to the clinical trials. And in clinical trials there are further 4 phases. Now what will happen in these different phases? In the first phase, the healthy subjects, healthy people are selected. And in healthy people, you will check whether the effects you have observed in the animals, you observe the same effects in the healthy people or not. And then you will check the dose response relationship, whether what concentration of your drug is producing how much response. So you do that kind of testing in the first phase. Then in the second phase, the testing in patients will be done. You will select certain number of patients and then you will do all the testings, and then you will check whether the desired effect is produced or not, whether it is producing the desired effect, that is correcting the disease or not. If it passes, if there are beneficial effects and adverse effects are small, then it passes the second phase and we move towards the third phase. In third phase, we take a larger group of patients and we compare this new drug with the standard drug which is given for that particular disease. We compare this new drug with the standard drug in the terms of therapeutic outcome. And if it passes this phase also, then you apply for approval of a new drug to FDA in USA and health protection branch drugs in Canada, UK and Europe. And then you if your drug is approved from those institutes, then your drug reaches into the fourth phase. And fourth phase is after marketing of the drug. When your drug is into the market, that is being prescribed and there is a widespread use of that drug in the population, then a regulatory surveillance is done. And during that phase also it is checked that whether it is causing any kind of adverse effects which were not observed during preclinical and clinical trials, and if those adverse effects are more as compared to the therapeutic effects, then that drug is withdrawn from the market. So let us discuss about the two main branches or two main divisions of pharmacology, that is pharmacodynamics and pharmacokinetics. In Greek, dynamic means power and kinesis means movement. Basically, what the drug will do to the body is pharmacodynamics. Whatever effect the drug is going to produce inside the body, what is the power of the drug, that is your pharmacodynamics. For example, we are administering adrenaline exogenously. It is going to act on the adrenocytes receptors or adrenoreceptors, which is our G protein coupled receptors, which we'll be discussing in detail. What they are going to do, that is what their mechanism of action is going to be, they are going to increase cyclic AMP, and they are going to cause cardiac stimulation, and there will be increased heart rate. So this effect is going to come, this drug will be producing this effect via this mechanism of action. This is the pharmacodynamics of the drug. What is kinetics? What the body will do to the drug once you have taken the drug. If you are taking the drug orally, for example a tablet, it will be disintegrated and broken down, and then it will be absorbed. After absorption, it will be distributed to different compartments of the body and basically your bloodstream. Then it will be metabolized in the body, mainly by your liver, and then it will be excreted or eliminated out of the body. For example, if you are taking paracetamol, it will be absorbed completely in your body, and your peak blood levels will be reached within 30 to 60 minutes, and it is 25% bounded by your plasma proteins, and it is widely distributed in the body. It is metabolized in the liver. It will be converted into inactive metabolites by glucuronide and sulfate conjugation, and after this conjugation it will be excreted into your urine. That is basically the pharmacokinetics, that how your body is going to treat with the drug. That is your kinetics. Now there are certain basic definitions which you need to know before we go into the details of pharmacology and details of different drugs. There are two types of drugs. There are essential drugs and there are orphan drugs. Now, what is the concept of essential drugs? These are defined by the WHO as those that satisfy the priority healthcare needs of the population. And if we define it in simple words, that is, which are essential, and these should be available to the general population easily. Because they are essential, they should be available to the general population, and if they are available to the general population, they have to be selected before they are being made available. So how they are selected? They are selected on the basis of their efficacy and their safety profile, their storage stability, their cost, and their effectiveness. And these drugs are selected, and in India there is a list of these drugs under the National List of Essential Medicines, which was formulated in 1996 and it was revised in 2003, and there are 354 medicines in that list. So this was the small concept of essential medicines. What are the orphan drugs? See, orphan drugs are the these are the drugs like any other drugs which are used for the diagnosis, prevention, and treatment of a disease. But in this case, this disease is a very rare disease. And because this is a very rare disease and very small population are using and consuming these drugs, therefore the profit the company is getting is very less. And there is no reasonable expectation of the company that the developing cost of the drug will be recovered from that profit, because the company is not getting profit, so they are not interested in the manufacturing of these drugs. But because these rare diseases do exist, these are uncommon but they do exist, so they need to be manufactured. So to encourage the companies to manufacture these drugs for the rare diseases, there is a act known as Orphan Drug Act in USA. There is a Orphan Drug Act in USA. In that act, what they offer is they offer tax benefits and certain incentives to these companies which manufacture these orphan drugs. So next we have to take pharmacology in detail. So we need to know how these drugs are going to react inside the body, what is their mechanism of action. So we need to know certain basic terms, and the first among them is receptors. So basically, what are receptors? These are glycoprotein molecules, and which are present on they may be present on the surface of the cell or inside the surface of the cell, but these are glycoprotein molecules which are, let us say, present on the surface of the cell. And what is their functioning? They recognize their ligand, and they bind to that chemical substance or the drug, and this will initiate a conformational change inside the receptor, and this will lead to a series of biochemical reactions which are translated into pharmacological effect. Basically, if you say a drug, when it is consumed, it get absorbed, distributed, metabolized, eliminated, everything. But when it gets absorbed into the bloodstream, into the systemic circulation, it comes in contact with its site of action, it gets binded to the receptor, and this binding will bring about a conformational change, and there will be series of biochemical reactions which ultimately lead to a pharmacological response. Okay, if we talked about receptor, there is a term known as spare receptors. So what are spare receptors? As the term indicates, spare means that something is extra which is present there. For example, there is a receptor here, there is a cell here, and it is having certain number of receptors, say 1, 2, 3, 4, 5, 6. It is having 6 receptors on its surface, and we are giving a drug, and suppose that drug molecule has covered these 4 receptors and it is giving the maximum response possible. Whatever concentration we have given, it has not occupied all the number of receptors, but it is giving the maximum response. And these two receptors are still available, but we have no more drug. We have already achieved our maximum response. So these extra free receptors, these are known as spare receptors. When maximum response is produced by the agonist at a concentration that does not occupy all the available receptors, the free receptors are known as the spare receptors. Okay, when we were talking about receptors, I used a term ligand, or some say it as ligands. So these are basically the substances which are coming and binding to a receptor. These ligands can be of two types. There are agonists and there are antagonists. Okay, agonists are the substances that come and bind to the receptor and they activate the receptor to give a pharmacological response which is similar to the pharmacological response which will be produced when a substrate is going to come and attach to the receptor. Now, what is a substrate? A substrate is an endogenous compound, an endogenous substance that is present inside the body, which is going to come and bind to the receptor and give a pharmacological response. This was our substrate. And what is an agonist? Agonist is a chemical substance which we are giving from outside the body. It is an exogenous compound which is given and it comes and binds to the receptor and it is giving the same pharmacological response as that of the substrate. Then it is known as the agonist. Okay. Now agonists can be of further different types on the basis of the extent of response they are giving. There are full agonists. Full agonists, as the name indicates, they are going to give the maximum response. There are partial agonists. Partial agonist means they are going to give a partial response. They are going to give a sub-maximal response. And there are inverse agonists. Inverse agonists are the compounds which come and bind to the receptor on that side and they are giving a opposite pharmacological response. For example, what is the response of the substrate? It is going to increase the hormone, but in case of inverse agonist, it is coming and binding and it is decreasing the hormone. That is the opposite reaction. That is your inverse agonist. Now, what are antagonists? Antagonists are the substances, they bind to the receptor and they block the activity of the receptor. They were enhancing the activity and antagonists are blocking the activity of the receptor. Further, they are of two types. There are competitive receptors, and there are non-competitive antagonists. In competitive antagonists, as the name indicates, these are going to compete. And compete for what? For the binding site. For example, this is the receptor, this is the substrate, and this is your antagonist. These two are going to compete, and whichever takes this place first is going to give the response. Suppose this comes and binds here before the substrate comes, and it comes here and it blocks that space for the substrate and blocking the activity of the receptor. This is a competitive type of antagonist. What is a non-competitive type of antagonist? As the name suggests, non-competitive, it is not going to compete. Simply what it is going to do is, there can be two types of non-competitive antagonism also. For example, what is going to happen is, either it is going to come and bind here and bring some of the conformational changes inside the receptor, so that the substrate, when it comes, it cannot bind to the receptor. Or what it is going to do is, it just has a allosteric site. This non-competitive antagonist will come and bind to this allosteric site or some other site, and it will bring about the changes in conformational changes in this receptor. Now the shape of this binding site, it changes, because of which this cannot enter inside the receptor, and thus the activity of the receptor will be blocked. So this is the non-competitive type of antagonism. Next important term which we are going to discuss is, whenever we are taking some drugs, they are going to have some effect on the body which can be addictive, or it can we can get dependent to that drug, or we can develop tolerance against that drug. These are the common terms which are used in pharmacology. So what does these terms mean? First is addiction. So addiction is a psychological or physical inability to stop consuming a chemical, drug, or activity or substance, even though it's causing you harm. You know that this drug is causing me harm, or it is having bad effects on my body, but still you are unable to discontinue the consumption of the drug. That is addiction, and it is producing symptoms inside your body. You are uncontrollably seeking for drugs, you are being secretive and hiding these drugs, and you are having relationship difficulties, you are having behavioral disturbances. These are some of the common symptoms which are faced or which can be seen inside a person who is suffering from addiction with any kind of drug or activity or chemical, anything. And what is the treatment of that addiction? Beside from the therapeutic or medical treatment, you can go for counseling. There are behavioral therapies, and there is ongoing care has to be uh, taken. The patient has to be taken care that he may not go into a relapse, because in addiction patients there is always a risk for relapse. Second term is dependence. Now, dependence is a physical condition in which body has adapted to the presence of the drug. That your body becomes used to that there should be drug present in your blood. And if that drug is absent from your blood, if the person who is repeatedly taking a drug and his body has become used to that drug, and he suddenly stops taking that drug, then he experiences withdrawal symptoms. And what are these withdrawal symptoms? He can suffer from anxiety, irritability, tremors and shaking of the body. There can be sweating, nausea, vomiting, loss of appetite. All these are withdrawal symptoms. And when your body gets used to and it is not getting that drug, then it will show you withdrawal symptoms. Third term is tolerance. Now, what is tolerance? Tolerance is that your body has tolerated a particular dose. When you are repeatedly taking a drug regularly, then the response of that particular concentration of the drug is reduced. There is a diminished or reduced response to a drug as a result of repeated doses. For example, the patients of chronic pain, they develop tolerance against analgesics. They become used to that particular concentration or that particular dose of that analgesic, that now that dose is not relieving them of pain. So in order to overcome tolerance, they have to increase their dose uh, so as to relieve their body from pain. The next term is tachyphylaxis. Now, these two terms are somewhat related, because tachyphylaxis is acute or sudden decrease in response, or you can say it is a short-term onset of tolerance. Tolerance was something which was occurring when there is repeated doses are taken of that drug. But tachyphylaxis is you have taken one initial dose, one single dose, and when you are taking that drug for the second time, suddenly there is a decreased response. Then you are taking the drug for the third time, the response is further decreased. So that is tachyphylaxis. That is a sudden short term onset of drug tolerance. For example, you took drug here. This is your effect. This is your time, and this is your administration of that particular dose. And you observed itna effect. Then for the second time you take the same dose, but the effect is reduced. Then you take the same amount of the dose, your effect is further reduced. So the sudden decrease in response to a drug after its initial administration, or rapid and short term onset of drug tolerance, that is your tachyphylaxis. And how the original response can be achieved again, how it can be restored, this can be done by increasing the dose now. So this was a lecture upon the introduction and the basic general pharmacological principles. In the next lecture, we will discuss about the pharmacokinetics and the ADME processes, that is absorption, distribution, metabolism, excretion, everything in detail. So that's it for now. Thank you.